My near and dear friends, welcome to this video session on reverse transcription polymerase chain reaction RT-PCR technique. What are the learning outcomes? At the end of watching this video, learner will be able to explain what is RT-PCR technique, what are the principle of RT-PCR technique, what are the advantage of PCR technique, and what are the limitation of RT-PCR technique. Introduction. Reverse transcription polymerase chain reaction occurs in two steps. First one, transcription, then second one, amplification and quantification. It is laboratory technique. In this one, we measure the amount of specific RNA. This is achieved by monitoring the amplification reaction using fluorescence, a technique called real-time PCR or quantitative PCR. Combined RT-PCR and qPCR are routinely used for analysis of gene expression and quantification of viral RNA in research and clinical setting. What is reverse transcriptase enzyme? The reverse transcriptase enzyme is discovered by Howard Tamin and his friend. However, the enzyme is first isolated in the year 1970 by David Baltimore. The enzyme is predominantly present into the retrovirus having the RNA as genetic material. The enzyme is RNA-dependent DNA polymerase or RNA-directed DNA polymerase present into the retrovirus. The retrovirus contains only three genes, GAG, POL, and the entire process of retroviral replication called reverse transcription. The combined RT-PCR and qPCR technique has been described as quantitative RT-PCR or real-time RT-PCR. In order to avoid confusion, the following abbreviations are used everywhere, such as polymerase chain reaction, abbreviated as PCR, reverse transcription polymerase chain reaction, RT-PCR, real-time polymerase chain reaction, qPCR, RT-PCR and qPCR in the combined form, qRT-PCR. This one is the reflection export. Before the introduction of RT-PCR technique, this technique was used for the RNA quantification. Now pause the video for a minute and write down the answer in your notebook. Now resume your video and check the answer. Reflection spot answer, that is Norton blot technique. Before the introduction of RT-PCR technique, this technique was used for the quantification of RNA. There are some limitations of Norton blot technique that are time consuming, require large quantity of sample, quantitatively inaccurate. Principle of RT-PCR technique. In RT-PCR, the RNA template is first converted into complementary DNA using a reverse transcriptase. Then complementary DNA is then used as a template for exponential amplification using PCR. The RT-PCR is broadly divided into two steps. First is the reverse transcription and the second is amplification and quantification. Principle is divided into two methods. One step RT-PCR and second one two-step RT-PCR. One step RT-PCR technique takes messenger RNA target and subject them to reverse transcription and then PCR amplification in a single tube. The setup of one-step RT-PCR is simple and easy. Accuracy and specificity of reaction is higher, therefore it is best choice for the screening and quantification assay. This one is the method. Select a one-step RT-PCR kit which should include a mixture with reverse transcriptase and the PCR system such as tech DNA polymerase and proofreading polymerase. Obtain all necessary material, equipment and instruments that are available in the kit. Prepare a reaction mixture which all include DNTP, primers, template, RNA necessary enzyme and buffer solution. Add this mixture to a PCR tube for each reaction, then add the template RNA. Place PCR tube in the thermal cycler to begin cycling. The first cycle is reverse transcription to synthesize complementary DNA. The second cycle is initial denaturation. During this cycle, reverse transcriptase is inactivated. The next 40 to 50 cycles are amplification program, which consists of three steps. First one, denaturation, annealing and elongation. The RT-PCR products can then be analyzed with the gel electrophoresis. This one is the pictorial diagram or pictorial representation of one-step RT-PCR. Advantage of one-step RT-PCR: this is the best choice for high throughput screening and for repeated quantification assay. The reaction is completed in the single tube, so the chances of contamination is very less. The reaction is fast and reproducible. What are the limitations? That are the major limitation of the one-step RT-PCR is the stock of complementary DNA. Here, the complementary DNA is directly synthesized into the reaction tube. Therefore, we cannot run two reactions separately for one sample type because the complementary DNA stock cannot be stored for further use. And second one, use of so many chemicals in one reaction. In the second two-step RT-PCR, as the name occurs in two steps. First one reverse transcription and second one PCR, amplification and quantification. This method is more sensitive than the one-step method. The setup of one combined template RNA primer and nuclease-free water in a PCR tube, add RNase inhibitor and reverse transcriptase to the PCR tube. Place PCR tube in thermal cycler for one cycle that include annealing, extending, and then inactivating reverse transcriptase. Proceed directly to PCR or store one ice until PCR can be performed. Second step, add a master mixture to the each PCR tube and add appropriate primer. Place PCR tube in thermal cycler for 30 cycle of amplification program, which include 3-step denaturation, annealing, elongation. The RT-PCR product can then be analyzed with the gel electrophoresis. This one is the pictorial diagram of two-step RT-PCR technique. Advantage of two-step RT-PCR. Advantage of two-step RT-PCR. First one, advantage, yes, for the smaller amount of sample, accuracy and specificity are higher. Third one, after the reaction complete, we have the complementary DNA stock for the second set of reaction. Here the two-step RT-PCR facilitate one additional benefit over one-step RT-PCR. We can store this complementary DNA for the further reaction. In the two-step RT-PCR, we are using 3 sets of primers, 1 set of oligo primer, 1 set of random primer and sequence specific primer. Hence the specificity of the reaction is higher. That is the limitation of two-step RT-PCR. The chances of the contamination and reaction failure is higher. It is time consuming. It is not used for high throughput screening. More expertization and additional optimization is required. This one is the procedure we will discuss in next video. The procedure include sample preparations, selection of primer, reaction preparation, RT-PCR cyclic conditions, strain synthesis. Advantage of reverse transcription PCR. The method can do a quantitative as well as qualitative analysis. A small amount of RNA can be used for gene expression study. Post-PCR processing such as agarosol electrophoresis is not required here. The method is simple, easy to use, rapid and cost effective. Also, the specificity and the sensitivity of the reverse transcription PCR is enormous. This one is a disadvantage of reverse transcription PCR. This method is extremely sensitive and even a small amount of DNA contamination can lead to false result. The method is restricted for some of the assay as higher expertization and experimentation are required to develop new assay. This one is the summary. Reverse transcription polymerase chain reaction is a laboratory technique followed by one-step RT-PCR or two-step RT-PCR. The method can do quantitative as well as qualitative analysis. This one is the reference. First one from Wikipedia. This one is the second one, genetic education. Thank you. Thank you for the watching.